Similarly, as we saw in the case of sucrose, when there is, very favorable conditions for the process of photosynthesis, for instance, in bright daylight, there will be more and more production of the carbohydrates. Specifically, more triose phosphates are produced, and these excess triose phosphates or excess carbohydrate production then can lead to the formation of more sucrose or starch molecules. Starch, as you know, is the major storage, carbohydrate storage molecule, or glucose storage rather, molecule in the plants. It is produced, it is stored and synthesized in plastids. It is made up of homopolymers, same type of homopolymers, same type of constituent units if it contains. Homopolymers of D-glucose, there is 1,4 linkage for the linear chains and alpha-1,6 linkage, alpha-1,6 linkage for the branch chain, as you have seen in the case of glycogen also. Glycogen, is the major glucose storage molecule in animals. Likewise, the major glucose storage molecule in plants is starch. It is stored for temporary basis in the chloroplast, and then for long-term basis in amyloplasts, in the seeds, in the roots, and in the tubers. As we saw and have been seeing again and again, whenever the monomers of a carbohydrate moiety are to be added sequentially, they have to be then attached with ADP through ATP, through ATP, or GDP through GTP, or UTP through UTP. Say these nucleotides are involved in binding with these monomers. Why? Because they make them high energy compounds. So, as was the case you saw in sucrose, as was the case you saw in glycogen, here also the glucose 1-phosphate is changed into ADP glucose, which will then be added up sequentially in the chain of the starch molecule, in the same way as you have seen in the synthesis of glycogen. So I won't go into the detail of it. A regulatory mechanism, it involves gene transcription, phosphorylation, certain redox conditions are involved in it. Then the key enzyme, the AGPase, ADP glucose pyrophosphorylase. This is one of the key enzymes which is involved in. Because why is it key enzyme? Because again, it's involved in the conversion of glucose into ADP glucose from ATP. There is a reducing agent known as dithiothriotol. It increases the starch synthesis. Why? It activates that key enzyme which is involved in the formation of ADP glucose. So in this way, it acts as an activating agent for the formation of starch.